The publication came out on the 22nd. It says here, the virus that causes COVID-19 has adopted some stealth moves to stay alive and kicking, and one secret to its success is hiding from the immune system by spreading through cell-to-cell transmission. A new study has found. Cell culture experiments showed that SARS-CoV-2, which causes COVID-19, limits the release of viral particles that can be inactivated by antibodies, instead staying tucked within cell walls and spreading between cells. It's basically an underground form of transmission, said lead author Shanlu Liu, a virology professor in the Department of Veterinary Biosciences at The Ohio State University and an investigator in the university's Center for Retrovirus Research. Liu and colleagues found other revealing details about SARS-CoV-2. The spike protein on its surface alone enables cell-to-cell transmission, and yet the virus's primary receptor on target cells to which the spike binds is not a necessary part of the cell-to-cell transmission operation. Additionally, they found that neutralizing antibodies are less effective against the virus when it spreads through cells. The research was published December 22, 2021, in the Journal Proceeding of the National Academy of Sciences. A major point of the study was comparing SARS-CoV-2 to coronavirus behind the 2003 SARS outbreak, known as SARS-CoV. The finding helped explain why the first outbreak led to much higher fatality rates and lasted only 8 months. We're about to surpass the 2-year mark of the current pandemic, with a majority of the cases being asymptomatic, Liu said. The comparison showed that SARS-CoV that caused SARS in 2003 is more efficient than SARS-CoV-2 at what is called cell-free transmission, when freely floating viral particles infect target cells by binding to a receptor on their surface, but also remain vulnerable to antibodies produced by previous infection and vaccines. SARS-CoV-2, on the other hand, is more efficient to cell transmission, which makes it harder to neutralize with antibodies. The virus's differing efficiencies were first demonstrated in experiments using pseudoviruses, a non-infectious viral core decorated with both kinds of coronavirus spike proteins on the surface. The spike protein is necessary and sufficient for both SARS-CoV-2 and SARS-CoV cell-to-cell transmission because the only difference in these pseudoviruses were the spike proteins. Looking more deeply into these differences, the researchers found that SARS-CoV-2 is also more capable than SARS-CoV at initiating fusion with a target cell membrane, another key step in viral entry process, and that stronger fusion action was associated with the virus's enhanced cell-to-cell transmission. Paradoxically, too much cell membrane fusion leads to cell death and actually interfere with cell-to-cell transmission. The team then turned to the role of the ACE2 receptor, a protein on cell surfaces that acts as a gateway for entry of the virus that causes COVID-19. The researchers found unexpectedly that cells with no or low levels of ACE2 on their surfaces can be penetrated by the virus, enabling robust cell-to-cell transmission. Finally, in experiments testing blood samples from human COVID-19 patients against the authentic SARS-CoV-2 virus, researchers determined that the virus could evade an antibody response through cell-to-cell transmission, but that antibody neutralization of the virus in cell-free transmission mode was effective. We were able to confirm cell-to-cell transmission is not sensitive to inhibition from antibodies from COVID patients or vaccinated individuals, Liu said. Cell-to-cell transmission resistance to antibody neutralization is probably something we should watch for as SARS-CoV-2 variants continue to emerge, including the most recent, Omicron. In this sense, developing effective antiviral drugs targeting other steps of viral infection is crucial. There are still many unknowns, including the exact mechanism the virus uses to spread from cell to cell, how that may influence individuals' responses to viral infection, and whether or not efficient cell-to-cell transmission contributes to emergence and spread of new variants. Lau's lab is planning additional studies using authentic virus and human lung cells to further explore these questions. This work was supported by grants from the National Institutes of Health and funds from an anonymous private donor to Ohio State. Shunlu Liu is also a program director of the Viruses and Emerging Pathogens program at Ohio State's Infectious Disease Institute.